Oligonucleotide Synthesis, Methods and Technologies, narrated by Luke McLaughlin, biotech digital marketer, business developer, and life science content creator. Oligonucleotide synthesis refers to the chemical process of creating short sequences of nucleotides, which are the building blocks of DNA and RNA. This process is fundamental in molecular biology, genetics, and biotechnology for various applications including diagnostics, therapeutics, and research. Basics of oligonucleotides. Oligonucleotides are short DNA or RNA molecules, typically ranging from about 5 to 100 nucleotides in length. They are used in genetic testing, research, and as therapeutic agents. Synthesis methods. Current DNA synthesis technologies and methods. 1. Phosphoramidite Method, chemical synthesis. This is the most common and established method for synthesizing short DNA sequences known as oligonucleotides. The process involves the sequential addition of nucleotide residues to a growing DNA chain, where each addition is protected by a phosphoramidite group. The method is highly efficient for generating oligonucleotides up to about 200 bases long. 2. Microarray-based synthesis. This technology allows for the simultaneous synthesis of a large number of different oligonucleotides on a solid surface. DNA sequences are built up in parallel on a microchip, enabling the production of thousands to millions of unique sequences at once, which are useful for applications like genome-wide experiments and large-scale synthetic biology projects. 3. PCR-based amplification. Often used to generate larger quantities of DNA from a small initial sample, PCR, polymerase chain reaction, amplifies a specific DNA sequence using cycles of temperature changes and enzyme activity. While primarily an amplification method, PCR can also be used creatively to synthesize new DNA sequences through techniques like overlap extension PCR. 4. Enzymatic DNA synthesis. This emerging technology uses template-independent DNA polymerases to synthesize DNA molecules. It represents a more natural approach compared to chemical synthesis, potentially allowing for longer and more complex DNA sequences to be constructed with fewer errors. 5. Terminal deoxynucleotide transferase, ITT, synthesis. This enzyme adds nucleotides to the 3' end of a DNA molecule without needing a template. TDT is used in specialized applications such as adding random or defined sequences to the ends of DNA strands in immunology and molecular biology research. 6. Gene synthesis via assembly of overlapping oligonucleotides. This method constructs entire genes from shorter, overlapping synthetic oligonucleotides. The oligos are designed to anneal to each other based on overlapping regions, and then are enzymatically ligated or assembled via PCR to form a full-length DNA sequence. This technique is essential for synthesizing complete genes and even whole genomes from scratch. Phosphoramidite oligonucleotide synthesis. Phosphoramidite oligonucleotide synthesis is the most widely used method for producing synthetic DNA and RNA sequences due to its efficiency and the high purity of the resulting oligonucleotides. This chemical method is based on the sequential addition of nucleotide monomers to a growing oligonucleotide chain, anchored to a solid support. Here's a detailed look at the biochemistry and specific methods involved in phosphoramidite synthesis. Biochemistry of phosphoramidite synthesis. Key components. Solid support. Typically a controlled pore glass, CPG, or polystyrene resin that holds the initial nucleotide fixed through a linker. Nucleoside phosphoramidites. These are the monomers used in synthesis. Each nucleoside phosphoramidite is protected to prevent unwanted reactions. The phosphorus atom is protected by a disopropylamino group. The 5-hydroxyl group of the ribose, or deoxyribose sugar, is protected by a dimethoxytrytyl DMT group. Additional protecting groups may protect other reactive sites, like exocyclic amines, depending on whether the sequence is RNA or modified DNA. Reaction steps: detritylation, removal of the DMT group from the 5' end of the anchored nucleotide, making the hydroxyl group available for reaction. Coupling. Addition of the next nucleoside phosphoramidite, activated by a tetrazole, to form a phosphite triester linkage with the 5-hydroxyl group of the previously attached nucleotide. Capping. Any unreacted 5-hydroxyl groups are capped with acetic anhydride and N-methylimidazole to prevent extension in the next cycle, ensuring that only fully extended chains react in subsequent steps. Oxidation. The phosphite triester linkage is oxidized to a more stable phosphate triester using iodine in water cypyridine solution. Methods of phosphoramidite synthesis. Stepwise process on solid support. Synthesis. Initiation. The synthesis begins with the attachment of the first nucleotide to the solid support. This initial nucleotide is already DMT protected at the 5' end and has a linker molecule that covalently attaches it to the support. Chain elongation. The cycle of detritylation, coupling, capping, and oxidation is repeated for each nucleotide added to the chain. The sequence is built in the direction from the 3' to 5' end, opposite to the natural biological direction of synthesis. Cleavage and deprotection. Once the desired sequence length is achieved, the full oligonucleotide is cleaved from the solid support. Protective groups are removed from the bases and the phosphate backbone in a final deprotection step, often using concentrated ammonia or methylamine. Automation. The entire process is typically automated in a DNA synthesizer, which precisely controls reagent delivery, reaction times, and environmental conditions, leading to high efficiency and throughput. Advantages and challenges. Advantages: high purity and yield. The solid phase approach allows for easy washing between steps, effectively removing excess reagents and byproducts. Scalability. The method is highly scalable, suitable for everything from small research labs to industrial scale production. Challenges: length limitations. As the length of the oligonucleotide increases, the efficiency of each step decreases slightly, which can lead to lower overall yield and purity for very long sequences. Synthesis errors. Errors such as deletions or insertions can occur, although they are relatively infrequent with modern techniques and machinery. Phosphoramidite oligonucleotide synthesis remains the cornerstone of molecular biology, facilitating advancements in genetic engineering, diagnostics, and therapeutics by providing a reliable source of high-quality DNA and RNA oligonucleotides. Microarray-based oligonucleotide synthesis. Microarray-based oligonucleotide synthesis is a sophisticated technology that enables the simultaneous production of thousands of specific oligonucleotide sequences on a single solid substrate, such as a glass slide or silicon chip. This method is crucial for genomics, diagnostics, and high-throughput screening applications. Here's an in-depth look at the biochemistry and methods involved in microarray-based oligonucleotide synthesis. Biochemistry of microarray-based oligonucleotide synthesis. The underlying biochemistry of microarray synthesis relies on the stepwise addition of nucleotides to growing oligonucleotide chains fixed on a solid surface. Each nucleotide addition is carefully controlled to ensure sequence specificity and high fidelity. Key components. Solid support. Typically a glass slide or a silicon chip that is chemically treated to attach a linker molecule which acts as the initial anchoring point for oligonucleotide synthesis. Linker molecules. These are covalently bonded to the solid support and provide a reactive group for the initial nucleotide attachment. Nucleotide building blocks. These are usually in the form of nucleoside phosphoramidites, each protected by a photolabile group that prevents unwanted reactions until specifically deprotected by light. Methods of microarray-based oligonucleotide synthesis. Photolithographic synthesis. This method, popularized by companies like Affymetrix, uses light-directed synthesis, a technique inspired by semiconductor manufacturing processes. It involves photolithographic masks. These masks are used to selectively expose regions of the microarray to light. The exposure removes the photolabile protecting groups from the active growing chain in a predefined pattern. Sequential addition. Once the protecting groups are removed by light exposure, a new nucleotide, distributed, protected by another photolabile group, is added to the exposed areas. Unexposed areas remain inactive due to the presence of the protecting group. Repetition. The cycle of light exposure and nucleotide addition is repeated, with each cycle adding one nucleotide to the growing chains in the exposed areas. Inkjet technology. Another method uses inkjet printing technology to synthesize oligonucleotides on microarrays. Nucleotide. Deposition. An inkjet printer is used to precisely deposit nucleotide building blocks onto specific locations on the microarray surface. Each nucleotide is again protected by a photolabile group. Site-specific synthesis. The process allows for the addition of different nucleotides to different spots on the array without the need for masks, making it highly flexible and customizable. Light activation. Similar to photolithographic synthesis, light is used to deprotect the nucleotides at specific locations where the next nucleotide is to be added. Advantages and challenges. Advantages: high throughput. Allows for the synthesis of thousands of different oligonucleotides simultaneously. Customization. Each spot on the microarray can be customized to carry a different oligonucleotide sequence, which is ideal for applications requiring a high degree of customization like DNA microarrays, SNP genotyping, and more. Challenges: complexity and cost. The equipment and technology required for microarray-based synthesis are complex and can be costly. Yield and purity. The yield and purity of oligonucleotides can vary, and the sequences are generally shorter than those produced by conventional in-solution methods. Microarray-based oligonucleotide synthesis has significantly advanced the field of genomics and molecular biology by enabling rapid and efficient analysis of gene expression and genetic variation. Its continued development and integration into research and diagnostic tools are pivotal for future discoveries and applications in the life sciences. PCR-based amplification. PCR-based amplification is a widely used method for generating large quantities of specific DNA sequences, including oligonucleotides, and for minimal initial samples. While primarily known as a method for amplifying DNA, PCR, polymerase chain reaction, can also be adapted for the synthesis and modification of oligonucleotides, especially when generating variants of a given sequence or replicating a targeted DNA segment. Here's an overview of the biochemistry involved and the methods used in PCR-based amplification for oligonucleotide synthesis. Biochemistry of PCR-based amplification. Key components. Template DNA, the sequence that contains the region of interest which needs to be amplified or modified. Primers, short oligonucleotide sequences that are complementary to specific regions at the start, 5' and end, 3' of the target DNA segment. These are necessary for initiating DNA synthesis. DNA polymerase, a thermostable enzyme that synthesizes the new DNA strands. Taq polymerase is the most commonly used, but other enzymes like PFU or fusion are chosen based on the need for higher fidelity or different properties. Nucleotides, DNTPs. The building blocks of DNA consisting of deoxyadenosine, deoxycytidine, deoxyguanine, and deoxythymidine triphosphates. Buffer and MgCl2. These provide the necessary ionic strength and pH for optimal enzyme activity. Reaction steps. Denaturation. The double-stranded DNA template is heated to around 95 degrees Celsius to separate it into two single strands. Annealing. The reaction temperature is lowered, typically between 50 degrees Celsius and 65 degrees Celsius, to allow the primers to bind and anneal to their complementary sequences on the template DNA. Extension. The temperature is adjusted to optimal conditions for the DNA polymerase, usually around 72 degrees Celsius, to synthesize new DNA strands, starting from the primers and using the original DNA strands as templates. Methods of PCR-based amplification. Standard PCR. The most basic form of PCR, it involves repeating the three main steps. denaturation, annealing, extension for 25. 35 cycles to exponentially amplify the target DNA segment. Real-time PCR, qPCR. Similar to standard PCR, but includes a fluorescent marker that allows for the monitoring of the amplification process in real time. It's used for quantitative measurements as well as synthesis. Nested PCR. Involves 2 rounds of PCR to increase specificity. The second set of primers anneals within the product of the first PCR, making it highly specific to the target sequence. Assembly, PCR or polymerase cycling assembly, PCA. Used for synthesizing long DNA sequences by overlapping multiple oligonucleotides. In the initial cycles, fragments are synthesized and overlap. In subsequent cycles, these fragments are assembled into a full-length sequence. Mutagenesis PCR, used to introduce mutations into a DNA sequence. Primers containing mismatches are used to incorporate the mutations during the amplification process. Advantages and challenges. Advantages, efficiency and speed. Large amounts of DNA can be synthesized in a few hours. Flexibility, can be adapted to synthesize variants, introduce mutations, or create new DNA constructs. Low starting material. Requires only minimal amounts of template DNA. Challenges. Error rate. Tag polymerase and some other polymerases lack proofreading activity, leading to errors in the synthesized DNA, though high-fidelity enzymes can mitigate this. Product size. Limitation. Longer sequences can be problematic due to enzyme processivity limits and the accumulation of byproducts. PCR-based amplification for oligonucleotide to DDCB synthesis is an essential tool in molecular biology, providing a versatile method for not only amplifying DNA, but also engineering DNA molecules with specific sequences and modifications. This method is integral to cloning, gene expression studies, and genetic testing. Enzymatic synthesis. Enzymatic DNA synthesis is an emerging technology that offers a powerful alternative to traditional chemical synthesis methods for oligonucleotide production. This method leverages the natural processivity and fidelity of DNA polymerases to synthesize DNA sequences, and it is particularly useful for incorporating modified nucleotides and producing longer strands with higher fidelity. Here's an overview of the biochemistry involved and the methods used in enzymatic DNA synthesis for oligonucleotide production. Biochemistry of enzymatic DNA synthesis. Key components. DNA polymerases. These are the enzymes that catalyze the synthesis of DNA molecules from nucleotide triphosphates, using single-stranded DNA as a template. Different types of polymerases may be used depending on the desired fidelity, speed, or ability to incorporate modified nucleotides. Nucleotide triphosphates, NTPs. These are the substrates that DNA polymerases use to synthesize DNA. In enzymatic synthesis, both standard, DATP, DCTP, DGP, DTTP, and modified nucleotides, for example, those containing labels or unusual bases, can be used. Template strand, a single-stranded DNA that guides the sequence of the newly synthesized strand. This can be a naturally occurring sequence or a synthetically designed oligonucleotide. Primers, short DNA fragments that are complementary to a specific region on the template strand. These are required to initiate DNA synthesis, as DNA polymerases can only add nucleotides to an existing 3' end. Reaction steps, primer annealing. The primer binds to its complementary sequence on the template strand. Chain elongation. The DNA polymerase adds nucleotides to the 3' end of the primer, extending the DNA chain in a 5' to 3' direction, complementary to the template strand. Termination. Synthesis continues until the polymerase reaches the end of the template or is stopped by other means, such as modified nucleotides designed to halt synthesis. Methods of enzymatic DNA synthesis. terminal deoxynucleotide transferase, TDT. Synthesis. TDT is a template-independent polymerase that can add nucleotides randomly at the 3' end of a DNA molecule. This method is useful for adding random sequences or labels to the ends of DNA strands. Rolling circle amplification, RCA. RCA is a method that uses a circular DNA template and a DNA polymerase to synthesize long, repeating sequences of the circular template. It is highly efficient for amplifying small circular templates into large quantities of concatenameric DNA. Polymerase chain reaction, PCR. While typically used for DNA amplification, PCR can also be adapted for synthesizing specific DNA sequences by carefully designing primers and controlling the cycling conditions. Polymerase cycling assembly, PCA. PCA uses multiple overlapping oligonucleotides and a DNA polymerase to assemble a longer DNA sequence from shorter fragments. This method is especially useful for building complex DNA constructs from smaller synthetic oligonucleotides. Advantages and challenges. Advantages: high fidelity. Enzymatic synthesis can achieve higher accuracy than chemical synthesis, particularly with high-fidelity polymerases that have proofreading abilities. Flexibility in incorporating modifications. Enzymes can often incorporate modified nucleotides more efficiently than chemical methods, which is beneficial for creating labeled or functionalized DNA molecules. Scalability and efficiency. Enzymatic methods can be scaled up and can be more cost-effective for certain applications, particularly when high fidelity or specific modifications are required. Challenges. Enzyme limitations. The choice of enzyme can limit the types of modifications that can be incorporated, and some enzymes may have difficulty synthesizing through certain sequences or structures. Complexity of setup. The need for a primer and a template can make the setup of enzymatic synthesis more complex compared to some chemical methods. Sequence limitations. Some polymerases have biases or difficulties with certain sequences, which can limit the complexity of the DNA that can be synthesized. Enzymatic DNA synthesis is rapidly advancing and holds significant potential for revolutionizing the field of synthetic biology and oligonucleotide production, offering a robust alternative to traditional chemical synthesis. Terminal deoxynucleotide transferase, TDT, synthesis, terminal deoxynucleotide. Transferase, TDT, synthesis is a unique and valuable method in molecular biology for oligonucleotide synthesis, particularly useful for adding non-templated nucleotides to the 3' ends of DNA molecules. This method capitalizes on the properties of TDT, a template-independent DNA polymerase that catalyzes the addition of deoxynucleotides to the 3' end of a DNA strand. Here's a detailed look at the biochemistry and specific methods involved in using TDTE for oligonucleotide synthesis. Biochemistry of TDT synthesis. Key components. Terminal deoxynucleotide. Transferase, TDTT. TDT is a DNA polymerase that does not require a template strand to guide its activity. It is unique in its ability to add nucleotides one at a time to the 3' hydroxyl end of a DNA molecule. Nucleotide triphosphates, NTPs. TDT can incorporate a variety of deoxynucleotides, including standard DNTPs, DATP, DCTP, DHDP, DTDP, and modified nucleotides, fluorescent biotinylated or otherwise labeled DNTPs. Buffer system, a specific buffer system that provides optimal conditions for TDT activity, typically including a divalent cation like co-circumflex 2+ or MN-circumflex 2+, which is essential for the enzymatic activity. Enzymatic reaction. Nucleotide. Addition. TDT adds nucleotides to the 3' end of an existing oligonucleotide in a non-templated manner, which means the sequence of the additions is random unless the reaction is controlled to add specific nucleotides. Reaction conditions. The reaction can be manipulated by altering the concentration of specific nucleotides, the divalent cation type and concentration, and the reaction time to achieve desired modifications or extensions of the oligonucleotide. Methods of TdT synthesis and labeling of DNA fragments. TdT is frequently used to label the three ends of DNA fragments with fluorescent or radioactive markers. This is particularly useful for applications like adding a tag for molecular probes, facilitating detection in various assays. Synthesis of randomized DNA libraries. By adding a mixture of all 4 standard dNTPs, TdT can be used to generate a library of DNA molecules with random sequences at their ends. This is useful for applications in protein binding studies where diverse DNA sequences are required. Tailoring oligonucleotides. TdT can also be used to add homopolymeric tails, for example poly-A tails, to the 3 end of DNA molecules. This tailing is beneficial for techniques such as cloning into T vectors or enhancing the stability of oligonucleotides. Advantages and challenges. Advantages: versatility. Able to incorporate a wide variety of modified nucleotides. Utility in labeling. Ideal for end labeling DNA strands with markers for easy detection. Customization. Facilitates the creation of DNA with customized sequences or chemical modifications at the 3 end. Challenges: randomness. The lack of template direction can be a limitation when specific nucleotide sequences are required. Dependency on conditions. The activity of TdT can be sensitive to reaction conditions, requiring careful optimization. Cost: enzymatic methods, including the use of specialized nucleotides and enzymes like TdT, can be more expensive than some chemical synthesis methods. TdT synthesis is a powerful tool for modifying and labeling DNA oligonucleotides with significant applications in research, diagnostic assays, and potentially therapeutic developments. Its template-independent mechanism offers unique opportunities for the creation and modification of DNA that are not easily achievable with other synthesis methods. Gene synthesis via the assembly of overlapping oligonucleotides is a fundamental technique used in synthetic biology for constructing entire genes from scratch. This method relies on creating a series of short synthetic DNA fragments, oligonucleotides, that overlap each other at their ends, facilitating their assembly into longer DNA sequences. Here's an overview of the biochemistry and the methods involved in this process. Biochemistry of gene synthesis. Key components: synthetic oligonucleotides. These are chemically synthesized DNA fragments, typically 40, 60 nucleotides long. They are designed to overlap each other by approximately 20, 30 nucleotides at their ends. DNA polymerase, an enzyme that synthesizes new DNA strands by reading the existing strands as templates. It extends the overlapping regions, joining the oligonucleotides into longer fragments. DNA ligase, an enzyme that seals nicks in the DNA backbone, creating a continuous DNA strand by covalently linking the phosphate backbone of adjacent nucleotides. Template DNA, optional. Depending on the method, a template strand might be used to guide the assembly, especially in methods that include PCR. Reaction steps. Annealing. The overlapping oligonucleotides are mixed and heated to denature them, followed by gradual cooling to allow the complementary strands to hybridize at their overlapping regions. Extension and ligation. DNA polymerase extends the 3 ends of the annealed oligonucleotides across the overlaps. DNA ligase then seals the nicks, forming a continuous DNA strand that comprises the entire gene sequence. Methods of gene synthesis, Gibson assembly. This method involves mixing all the components, oligonucleotides, DNA polymerase, DNA ligase, and an exonuclease, in a single reaction. The exonuclease chews back DNA ends in a controlled manner to create overhangs that enhance the annealing and joining of fragments. Golden Gate assembly utilizes restriction enzymes and DNA ligase. The oligonucleotides are designed with specific sequences compatible with a set of restriction enzymes. These enzymes cut the DNA to create sticky ends that facilitate the directional and ordered assembly of the fragments. S-Elig, sequence and ligation independent cloning. Similar to Gibson assembly but does not require a ligase, the exonuclease activity creates overhangs that naturally anneal, and the gaps are filled in by DNA polymerase during a subsequent PCR step. Polymerase cycling assembly, PCA, uses overlapping oligonucleotides and cycles of denaturation, annealing, and extension with a DNA polymerase to progressively assemble the entire gene. PCR is often used in final steps to amplify the full-length product. Advantages and challenges. Advantages: accuracy allows for precise control over the gene sequence, enabling the inclusion of specific mutations, tags, or regulatory elements. Flexibility, adaptable to almost any sequence, regardless of length or complexity. Scalability, suitable for assembling multiple genes or large constructs simultaneously. Challenges: design. Complexity, requires careful design of overlapping regions to ensure specificity and efficiency of assembly. Error propagation. Errors in the initial oligonucleotides due to synthesis inaccuracies can propagate through the assembly process. Optimization required. Reaction conditions, for example, temperature, ion concentrations, enzyme mix, often need optimization for different sequences or lengths. Gene synthesis via the assembly of overlapping oligonucleotides is a cornerstone of modern synthetic biology, providing a powerful tool for creating novel genetic constructs. This method has enabled significant advancements in gene editing, synthetic genome projects, and the development of customized genetic pathways for industrial and therapeutic applications. Conclusion. In conclusion, the synthesis of oligonucleotides, a crucial component of modern molecular biology and biotechnology, encompasses a variety of sophisticated methods, each tailored to meet specific scientific needs and applications. From the highly efficient and widely used chemical Phosphoramidite Method, which excels in producing high-purity oligonucleotides quickly, to the precision of enzymatic synthesis, which allows for the incorporation of modified nucleotides and higher fidelity. These techniques highlight the versatility and adaptability of oligonucleotide synthesis technologies. The photolithographic and microarray-based approaches have revolutionized the ability to produce vast arrays of oligonucleotides simultaneously, thereby accelerating genomic research and customized diagnostics. Meanwhile, PCR-based amplification techniques have become indispensable for their ability to quickly and efficiently amplify DNA sequences from minimal starting material, facilitating numerous applications in research, diagnostics, and therapeutic development. Further, specialized methods like terminal deoxynucleotidyl transferase (TDT) synthesis offer unique capabilities in adding non-templated nucleotides to the three chelacein ends of DNA molecules, proving essential for specialized tagging and labeling of DNA. Lastly, the assembly of overlapping oligonucleotides through techniques such as Gibson assembly and polymerase cycling assembly showcases the intricate choreography of DNA assembly, enabling the creation of large DNA constructs and entire synthetic genomes with remarkable precision. Each of these methods contributes uniquely to the field, demonstrating the robustness and innovation driving oligonucleotide synthesis. As these technologies continue to evolve and improve, they promise to unlock further potential in genetic engineering, therapeutic development, and beyond, highlighting the dynamic interplay between scientific inquiry and technological advancement in the pursuit of understanding and harnessing the power of nucleic acids. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about these topics, consider reading our full-length articles on biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.